deals with our adaptive immune response. And here now we're talking about antigen-antibody interplay, if you will, interaction. Our whole adaptive immune response is based on the antigen-antibody response. So the two main players here are the B cells and the T cells. And we identified humoral immunity versus cellular immunity. Humoral, another word for that is liquid. So you can think of humoral immunity as being liquid immunity. That's the B cells. And the B cells are then, in our circulatory system. That's where we're going to see their response. The T cells then deal with the cellular immunity. So they're reading our cells. The helper T cells and the cytotoxic T cells actually are coming in contact with cells that they're responding with. They don't come in contact or deal with free antigens at all. They have to have direct contact with B cells or direct contact with macrophages, or in the case of cytotoxic T cells, direct contact with body cells or whatever they're dealing with. They have to come in contact with. So you have to have that interaction between surface proteins and surface receptors in order for these cells to interact. Yes?
Do T cells also interact with the dendritic cells?
Yes, they do. So if we think about B cells now, their role first, they are encountering antigens. Now you remember, every B cell is able to recognize only one antigen. And so we can have 100 million different B cells able to recognize 100 million different antigens. So the right B cell has to come in contact with the right antigen. This is where sitting in the lymphatic system, sitting in one of those lymph nodes, letting the bacterial products wash over you improves your chances of getting a B cell in contact with the right antigen. So now the B cell has come in contact with the right antigen. It's going to internalize that antigen. It's going to bring it in. And if you remember, it's going to break it up into different parts. And now it's going to present those parts onto the surface, waiting for a T cell to come along and recognize any one of those antigens that it's presenting here. Now, keep in mind that the antigen that the B cell initially recognized that caused it to internalize that microbe or whatever is not the same antigen that it is presenting on the surface now to T cells. It could be one of any number of different antigens on the surface that the T cell will recognize. So one of the ways to think about this is that this is our B cell, and it has, a receptor for an antigen that will fit here perfectly. So it binds to that antigen, brings that antigen inside, internalizes it, and then takes this antigen and breaks it up into parts. I should use a different color here. Breaks it up into parts. Like so, and then these parts, if you remember, are going to be presented on MHC molecules. So you can see, even though the B cell is recognizing only one antigen, it is potentially presenting several different antigens to a T cell, because it's breaking this antigen up and presenting parts now. Now, a T cell comes along and recognizes this specific antigen here. Should make it that way. So if that T cell recognizes that antigen, it can stimulate this B cell to expand clonal expansion that we talked about. And all B cells then will still have this same receptor on the surface to recognize this antigen. Even though the T cell is responding to this being presented, it is this B cell, or it's this antigen that the B cell is responding to. And so it is this antigen that the B cell will produce antibodies for. Okay, so now the T cell, if it's recognizing this antigen being presented, it will release cytokines, stimulate this B cell to undergo clonal expansion. Then it'll undergo differentiation into plasma cells, and then those plasma cells will start antibody production. Go ahead.
So is a plasma cell a type of B cell?
Yes.
Okay.
It's a differentiated B cell that is,
It's going to produce antibodies.
and that's all it does. Okay, so that's so late in the infection now.
Right.
I mean, you're days into the infection when the plasma cell shows up, because if you remember, it takes 10 to 14 days to get antibody production up there after initial exposure. And the reason is because this process takes so long. So we've only talked about the end of it here. You have to remember now the T cell here is only able to recognize this antigen that's being presented to it because of the dendritic cell that earlier had presented that same antigen to it. So this dendritic cell, who also likely had a receptor for this antigen, picked it up, brought it in. Maybe it wasn't specific. Maybe it was just a toll-like receptor that bound here, but it was enough that it brought it in. Now, because the dendritic cells, they're non-selective. They're going to engulf anything really that binds to their toll-like receptors. So they bring it in, but now they're presenting it on, they've internalized it, broke it up, and they're presenting a part of it now to the T cell. This is happening in the lymphatic system. So the dendritic cell has moved into the lymphatic system and is presenting this to the T cell. If the T cell has the appropriate surface receptor for it, so that it recognizes it, that can serve to turn it on, except for the dendritic cell has to be releasing what we call co-stimulatory molecules. See, those co-stimulatory molecules, they're going to diffuse over to the T cell, and they're going to tell the T cell that this antigen that you're recognizing comes from a bacterium, or comes from something of concern. So without the co-stimulatory molecules, the T cell likely will ignore this antigen, and if presented again, would just ignore it completely. In fact, this T cell might then eventually degrade. So once presented with the antigen from the dendritic cell, the T cell now becomes active, an effector T cell, if you will. And so if it does get presented the antigen by the B cell, it can release cytokines that will diffuse over to the B cell and trigger the response. Now recognize this B cell has surface receptors for each of these cytokines. And so that's how you get your reaction.
Are those helper T cells, or are they just generic?
These are helper T cells, yes. Yes.
Question.
Sure.
So, I know there's two classes of MHC.
MHC molecules, yes.
So the first class, is that the one that, I guess I'm trying to say, is that one due to the helper T cells, or is that one for the
It's MHC class 2 that hook up with the T helper cells, right, Mark?
Yes.
Yes, thank you. So let's put that up here. T helper recognizes the MHC class 2 molecules, and these class 2 molecules are found on antigen presenting cells. Okay, so B cells, macrophages, and dendritic cells have MHC2 molecules. And so the T cytotoxic cells, they recognize MHC1 molecules. These are molecules that are found on body cells, somatic cells. This is a way of determining whether or not that cell maybe has been infected by a virus, because it's sampling internal structures and presenting those to the cytotoxic T cell. If it recognizes something that tells it the cell's been infected, it will release cytokines or molecules and cause apoptosis, which is cell destruction from the inside. Remember me mentioning that? We don't want the cell to explode, lyse, rupture, because if it is filled with bacteria or viruses, we don't want those to spread into the circulatory system. So in this way, cytotoxic T cells release their chemicals and they cause the cell to destroy itself from the inside.
So is all this happening simultaneously?
Pretty much. I mean, you have to imagine that you've cut yourself, exposure's taking place there, your dendritic cells there are collecting the antigen and moving into your lymphatic system. At the same time, you've got fluids moving into the lymphatic system that are carrying the antigen that will bind to a B cell. Okay, so maybe that takes a day or two for that to happen. And then the dendritic cell does come in contact with the T cell and presents it. Okay, now the T cell is activated and it's moving around. Okay, eventually comes in contact with the right B cell that's now also presenting the same antigen. And only then do you start to get to antibody production. So, you know, we're talking 10 to 14 days from initial exposure to antibody production to the level that your body's going to be able to fight off the infection.
And the naive T cell, in its naive state, can really take up any antigen that the dendritic cell presents?
No, it is highly specific. It can only recognize a specific antigen. So if this T cell maybe doesn't recognize this antigen, it goes away, or excuse me, over here. Doesn't recognize this antigen, it goes away, and a different dendritic cell will come along and present a different antigen. So it's going to be very specific with the antigen that it's going to recognize.
And the dendritic cell, it internalizes and breaks it into pieces too. So it's getting those same pieces. So all it has to do is recognize one piece.
It will only recognize one piece. Even though the B cell may be presenting different pieces, this T cell is only going to recognize one. Oh, or not. Okay? But if it does recognize any, it will only be one.
And it'll only recognize after it's already come in contact with the dendritic cell.
Right.
And the stimulatory molecules.
Yes.
And the dendritic cell releases the stimulatory molecules at the same time as it's presented.
Yes.
Yeah.
The activated T cell, that is a helper T cell, right?
Yes.
But the naive T cell, is that also a helper T cell?
Yes.
Okay.
So you can have a helper T cell that is naive because it hasn't been presented an antigen yet. You can have a helper T cell that is an effector T cell because it has been presented. And you can have the same applied to cytotoxic T cells. You can have naive cytotoxic T cells because they too have to be presented an antigen. And you can have effector cytotoxic T cells that are already been activated.
So is that the reason you say in exposure you only have like some hours? Like,
Well, now with the second exposure, after all this has gone on, we've talked about plasma cells and the huge antibody production you've gotten from the plasma cells. In addition to the clonal expansion and differentiation, you're going to produce a few memory cells, a few memory B cells. And those memory B cells do not have to present to the T cell if they encounter the antigen again. They can undergo expansion and differentiation without the T cell regulating, without the T cell's approval. And so you have those B cells, and so if this antigen comes around again, we don't need this step. It can go right into antibody production upon a second exposure. And this is why you generally don't get sick from the same organism shortly after being exposed to it the first time, because your memory B cells allow for a quick response, your antibody titer jumps And so now all of the bacteria and stuff are coated with antibodies, and so they can't replicate. And so your immune system comes along, macrophages, eats them up and everything.
It's eating everything.
I'm sorry?
That's the reason the second exposure, that's when it's eating everything.
Yeah, well, you get massive antibody production quickly. So that antibodies bind to everything. Now, when the antibodies bind to it, even if the macrophages haven't had time to engulf them, they're not replicating now because the antibodies sort of cause them to, clump together, prevent them from replicating, essentially immobilize them. So they can't do their damage once bound by the antibodies. So that huge antibody production that we get from the second exposure within a couple of hours or days serves to pretty much tie up all of the bacteria and give our immune system a chance to go in and eat the bacteria and stuff.
I have one more question. You may
Sure.
have already said this, but the MHC molecules are presented, the MHC1 molecules are presented on what kind of cell?
Somatic cells are body cells. Okay, so anything that's not an antigen presenting cell.
And when the B cell initially recognizes the antigen, that is the process of clonal selection.
Yes.
And then when it internalizes it, each piece that it breaks up to present to a T cell, that's called epitope.